Good afternoon. It's been a long and exciting day, I hope, for everyone. And you've already heard mention, I think we're here to talk about parcellation. We're here to talk about how we can divide the brain up into distinct regions based on specific features, whether they're connectivity or functional activation patterns or cytoarchitectonics. And some folks in their prior talks have been alluding to the possibility that some of the divisions between regions are not as sharp as we might like, and the brain doesn't necessarily divide up into these sharply bounded entities that we can describe using multiple modalities. And in part, I did focus the title back when we designed this education course on functional connectivity, but what I'm going to be describing is really applicable to connectivity in general, and really beyond that, more to methods that have been emerging for trying to characterize the degree of transition between regions. Instead of trying to simply cluster and find a sharp boundary, to look at how features change over space. And to begin, we've talked about this indirectly, but there's a set of criteria that have been discussed in literature for how do we define a cortical area. And it's the FACT criteria, easy to remember. It's you got function, which is this feature having a unique response to specific stimuli or task demands. Architecture, so side or myellar architecture, distinct properties of the cortical microstructure within a specific region. There's connectivity, various modalities that we can use to measure that. And one that we haven't touched on as much, which is going to be the focus now of the talk, is how we can look at topography. It's kind of the which of these things is not like the other. We have these three criteria that are each more homogeneous within a cortical region than they are to their neighboring areas, except for topography, which is actually itself characterizing differences within a region. And I'll go into a few examples of that I think we're all aware of very much. But also in characterizing these differences in the function or the functional representation within a specific area, it also helps us to map out boundaries, because we can look at where those topographic maps get reversed, where lines of symmetry exist, and it's yet another feature that we can use to find the boundaries between specific regions. So it's complex in that way of both characterizing difference, but also representing difference across areas. Some examples that we're all familiar with from the classic texts of topographic representation or topographic maps include retinotopy, so the fact that we have representation of our visual information from the environment actually spatially represented continuously across primary visual cortex, somatotopy within both primary somatosensory as well as primary motor cortex, in which there's representation of the body schema across the cortex, moving from ventral to dorsal, and tonotopy within primary auditory cortex, in which we have frequency spectrums represented spatially across primary auditory cortex. In addition to what's been well characterized and really early findings within neuroscience, within human neuroscience, in addition to the observation of these topographic maps within primary areas, there are topographic maps which extend to association portions of the cortex, which extend beyond the boundaries of primary areas. And these include more complex processing, certainly based off of the topographic maps that are located within primary regions, but also broadening, becoming more distributed. And that includes features such as extrastriate retinotopy, saccade target angle, which is represented throughout the cortex in these patches that are presented here in the figure in the middle. And another example that we can talk about in exploring the question of topographic maps is one of how does connectivity vary across space. So up until now, I've looked mainly or discussed mainly how different features as they're represented in the environment or motor movements are spatially distributed. The last example on the far right is cortical striatal loops, and I put that in because I think it's a unique example of how does connectivity, which one can talk about having a rostral caudal gradient through the striatum, then get projected into rostral caudal gradients that are represented within, in this case, the frontal lobe. And so we can begin to talk about connectopies, or spectra of connectivity as they're represented over the space of the cortex and repeating throughout the cortex. So all these are examples of different topographic maps and basic organizing features that we can use to describe the spatial organization of the cerebral cortex. One thing that I haven't mentioned, and I've done that intentionally to keep this a bit conservative, is I haven't talked about the issue, and I won't be discussing that in the talk today because I didn't realize how much it would come up in the prior talks. I would have if I'd known. The issue of sharp versus gradual boundaries that I've mentioned. Because I think that, again, these methods are capable of addressing those questions of the degree of transition between cortical areas, but it is a distinct problem. And so instead I'm really focusing on this issue of topographic maps because it's certainly less controversial, but also an organizing feature of the cortex that is worth further investigating methods we can use to explore these features. And just to give another sense of why this might be valuable and just a couple examples of findings that I think are exciting for further work in characterizing how topographic maps relate to sensory impairment. This is a beautiful study by Sato and Grossman and colleagues that was published a couple of years ago looking at somatotopic reorganization following surgical intervention in spinal cord damage and reorganization that was observed in somatotopy. So what they did was put individuals in the scanner and they tactily stimulated across the body schema. Within healthy individuals on the left, you can see they observed a really nice gradient of somatotopic organization, which was disturbed in individuals with specific damage. Following surgical intervention, that gradient was relieved and returned to a more normal state. Another example of lines of work looking at somatotopic in this case, retinotopic organization in individuals with congenital blindness, in which using functional connectivity in this case, they were able to observe between healthy individuals and congenitally blind individuals similar retinotopic organization using functional connectivity in the primary visual cortex. However, when connectivity to association or areas of the cortex was explored across these two groups, there were differences that were found. So it was the relationship between primary visual regions and association areas that was found to be different between these groups. Now, specifically looking at topographies using functional connectivity, there's this interesting feature that's already come up in the discussions and the presentations before of what aspects of let's say cortical boundaries are best captured using resting state functional connectivity and various connectivity measures in general. And there's a nice review from Buckner and Yo in Neuroimage published a few years ago that discusses one of the differences that can be found between looking at global boundaries and local boundaries in functional connectivity. And so on the left in the middle image, you can see these nice boundaries that delineate V1 from V2. On the lips, these boundaries are on the lips of the calcarine sulcus, which delineate those areas cytoarchitectonically. And the image on the left in the center demonstrates a nice correspondence with the cytoarchitectonic boundaries between V1 and V2. This is a method that's much more similar to the Gordon, Cohen, Wig, Washiu, essentially, methods that are looking at local changes in connectivity patterns, and they're highly sensitive to these cytoarchitectonic boundaries. On the right, however, you're seeing the results of a set of methods that are more characteristic of what Buckner and Yo have been using, which are using global connectivity patterns to distinguish between regions. And if you're to draw a boundary using this map that we have on the right to delineate clusters, you would have one that's essentially segregating along the eccentricity, so peripheral from the focal regions there. And so one of the aspects that we can take into account when using functional connectivity as a way of mapping topographies is that the global patterns that we're able to use for feature extraction are going to be more sensitive to these topographic maps that are related to the environment. And that's what I'll be focusing on in the next methods that are presented. I want to show one other example of this using specifically methods that are related more to global patterns of connectivity. And this, I think, is a nice example because if the functional connectivity were to map onto cytoarchitectonic boundaries better, you would expect to see that as you move higher in the cluster number, there would be a distinction between primary motor and primary somatosensory cortex, so that the cluster boundary as you moved higher in the number would fall right along the central sulcus here. But instead, actually, what's being divided is along the line delineating somatotopic representation. And that it would continue to occur as we moved higher in the cluster number. It appears that as you use global connectivity patterns, the distinctions fall along these topographic maps within functional connectivity. Does that make sense, more or less? because it's an important feature to take into account. And yeah, so basically in summary on this point, it appears to be well tuned to distinguish between these topographic patterns. Okay, so to go back to like the simplest way that we could approach this problem, if we want to understand ways in which functional connectivity could be used to map topographic patterns along motor cortex, we simply take seed regions along the precentral sulcus here. So this is work by Martin van den Heuvel from about a decade ago now. Placing seed regions starting ventrally and moving dorsally. What we see is a nice change in the connectivity patterns that move more and more dorsally. As you would imagine, there's more connectivity to specifically adjacent regions as we move towards more dorsal portions of the cortex. And one of the observations from this study, which was one of the first ones to look at these subtle changes in pattern over the motor cortex, was that regions that are at the same position along this ventral to dorsal gradient are more interhemispherically or preferentially interhemispherically connected with one another. So you can see on the right these nice parallel lines, because as you move further from ventral to dorsal along the motor cortex, you get preferential connectivity to the contralateral side. And while this was a really clear and say already an impressive application of resting state fMRI and hadn't really been observed before in these patterns, it's only looking at one dimension of connectivity, a single line along the strip of cortex. And the next challenge would be, how can we find a method that allows us to pull out these patterns, these distinctions of connectivity patterns, without having to simply look at each of these individual maps? How can we characterize this in a data-driven way? And that's what Kuhn Hack, working with Christian Beckman, developed and published last year in Neuroimage. This is already the results to show you where this is going, and we're going to go a bit into the methods that are available to extract these patterns. They use the term Connectopic mapping because essentially these topographic maps are mapping consistent patterns of connectivity across the cortex. And here are the basic steps. So we extract connectivity from all the points within a region. So we can take connectivity from the precentral sulcus. We construct a similarity matrix of these connectivity patterns, and we can go into different versions of that in a moment. We then use spectral embedding to map the dimension that represents the most variance in these connectivity patterns, to essentially stretch out this connectivity graph to find patterns in connectivity. And these are referred to with some different terms that I'll also go into in a moment. And finally, we project that back into brain space to represent the variance in these patterns of connectivity. I'm going to go through these 4 steps in a couple of different ways because they're the core of these methods. And my hope is that by the end of this, one of those approaches for describing these different steps will click, and you'll be able to leave with an understanding of some of the basic methods that are used for representing these topographic patterns over the cortex. All right, so in prior talks, we've already heard a nice reference to Heidi Johansen-Berg's 2004 really seminal work looking at parcellation, in this case taking spectral reordering as the basic approach for looking at differences in connectivity pattern across the SMA, supplementary motor area, pre-SMA, along the medial wall of the cortex. And essentially taking a connectivity pattern represented here, reordering it so that connectivity patterns that are most dissimilar from one another are furthest away from each other. And what's important about this move of spectral reordering is that it allows one to then visually cluster, in this case, and to say, here's a sharp distinction between these 2 groups of patterns, which we can then project back into these clusters in the cortex, and allows one to represent the data in a way that we can make that decision of where to segregate the data, but also have visual information about the degree to which these patterns are really distinct from one another. One could have also imagined a result in which there was a smooth transition moving all the way along this diagonal, but instead you see that there's a sharp distinction. This is based on an approach called Laplacian eigenmaps, which are representing variance in the connectivity patterns. And along the lines of some of the discussion that's been taking place with respect to the insula, was first applied by Leonardo Serliani a few years ago in 2012 using diffusion-based probabilistic topography to map connectivity of the insula, and then to represent the degree of connectivity transition as you move through the insula rostral caudally here. And why this was important is that when Leonardo was first looking at ways to distinguish regions of the insula, he didn't find a sharp distinction using some of the classic clustering methods. It was difficult to derive what was the proper scale for describing different subdivisions. And so instead he went deeper into the methods and found that you could use this representation of dimensions within the connectivity matrix to then represent smooth transitions through the insula. So what are the methods for using spectral reordering? In this work by Claude Bachada, that was published last year in Neuroimage, they take probabilistic tractography maps from every voxel, in this case within the temporal lobe, extract the features. So we have seed voxels that we're interested in by the target voxels, which we've already discussed. We compute the similarity of each of those seed voxels. And then the spectral reordering step that we'll go into in more detail in a few moments. And as you can see here, there's not a clear boundary into two clusters, which is why, in this sense, it's reasonable to represent this spectrum of connectivity, this order of connectivity, this rank of different voxels along this dimension. Because what it's represented as we move along this matrix is differences in these connectivity patterns without a sharp distinction into different clusters. And then we can project that back into the cortex to say that areas in blue here are maximally different from those in red. And that's what we're going for in these patterns, and that's what all these methods are getting at. How can we talk about maximally different patterns in connectivity along this brain space? Okay. In bringing this back now to resting state functional connectivity data, one might ask the question of how we can represent multiple different gradients or multiple different patterns of change across the cortex in connectivity. One of the problems, of course, here becomes that if we do, in fact, have multiple different overlapping gradients of connectivity, they can get mixed up, they can get enmeshed, so that when we then essentially cluster the original data, we would be drawing distinctions between regions in an artificial way. And I'll show you what this means in the example of primary visual cortex. We have eccentricity and polar angle being represented within V1, in this example by Hawk and colleagues. We can extract out orthogonal dimensions of connectivity gradients, Connectopic 1 and Connectopic 2. But if there's a superposition of those and we draw a line delineating parcellation between these two halves, it's simply a mixture of the two gradients in connectivity that's an unrealistic representation of the data. And so the first step in trying to map these various gradients in a meaningful way is to pull out these orthogonal dimensions. The way that they do this, again, going through each step, is to start with a source region that they're interested in. So in this case, let's take the premotor sulcus to extract out data from that region of interest over time, and then to look at the correlation of that region to points outside of that specific ROI. So how those correlate with regions outside of the region of interest. We then create a similarity matrix, conduct a Laplacian eigenmap analysis, which gives us the connectomic maps at the bottom here. I should mention that all of this analysis has very recently been made available as of last night on GitHub. So we were going back and forth, and I was going back and forth with Cohn, who's a wonderful researcher at the Donders. He's just put this analysis up, so it's an exciting moment, and there'll be a talk on this method on Thursday afternoon. I'll put the exact time up at the end by Christian Beckman, who'll be presenting it in the Open Science Room. In addition, for an introduction into the Laplacian eigenmaps approach with respect to connectivity data, I highly recommend a review, or a summary, really an appendix, by Leonardo Serliani in his 2012 Human Brain Mapping paper, in which he goes into the specific application and motivations for this approach as it applies to connectivity data. Okay. But to give a basic introduction to this, how many folks, are folks here familiar with the term Laplacian eigenmap? Yeah? Okay, great. So, yes. Feel free to jump in here for those who are. For myself, and who isn't familiar with it? Great. Okay, I wasn't really either until relatively recently, and so I'm going to try and give you a very intuitive feel, a geometric feel for what this is doing and why. Because it was something we struggled with in the lab a lot. It seemed to make, everybody was into it for talking about these topographic gradients, but it was difficult to understand specifically what it was doing mathematically that was so important. And there are many ways to try and explain this. What I'm going to try and do, I was thinking as I was standing up there, what might we do? let's say if I was going to take a ball in one hand, this is a ridiculous example, but hopefully it'll make sense. If I'm going to take a ball in one hand and roll it, like a juggler kind of across my body over to the other hand, what's critical about that act of rolling a ball across is how far these hands are from each other over space. It's walking along this space from one arm to the other. If I have my hands folded in this way, and I'm talking about the distance between my two hands here, it's not relevant to that question of rolling a ball across. It's not relevant to how far it takes for that ball, let's say, to diffuse in space along my arms from one to the other. And so the first step that I want to do in measuring that distance is to unfold my arms and to stretch them out. The reason I'm giving you this example is because this is what I have to work with at the moment. But you can imagine the same problem with a cord that's tangled up. The first step, if we want to know how far away 2 points are along a string or a wire, is to unfold that wire, and then we can begin to talk about decomposing that space into something that's more a dimensionality reduction of that space. Same thing if we have a banana and we want to look at points along the banana. The first thing we want to do is to straighten it out, because what's relevant if you're walking from one point of banana to the other is how far those points are with respect to one another, not how the banana itself is embedded in the three-dimensional space that's irrelevant for walking along it. Yeah, please. Yes. Please, yeah. No, I mean, reducing the space to a combination of the local information, which is more relevant in this case, with the global information. And let's see if I did this correctly. Boom, yeah. It's essentially unfolding these complex spaces so that what's included in the calculation then, the dimensional reduction step, is based on these local relationships between points. It's walking along this space. Except in this case, it's not the body, it's not a banana, it's not a bunch of points that are rolled up like a Swiss roll, it's the connectivity graph. And so what we're doing is taking this big ball of connections between points in this matrix and stretching it out so that points that are maximally distant from one another actually get kind of unfolded and stretched out. A couple of sort of points to mention. There are multiple different normalization steps that one can conduct here. What they're essentially going to do is render the data more or less sensitive to noise. So if there are outliers, those would drive an analysis. Some of the normalization is going to minimize the impact of outliers in this graph space. So you can imagine that there's a point that's far over here along the Swiss roll. That would then, when you're doing the decomposition step, essentially a principal component analysis, that would then drive the variance and drive the eigenfunctions that are the result of this analysis. What's also important about this step is that it's about the probability of transition from one point to its neighbors. So it's not going to be driven by more central nodes in the graph. It's normalized at each point so that all of the rows sum to the same value. In practice, setting the alpha value here, the normalization value, to the exponent to 0.5 makes this more robust to outliers, and this is what we've been doing in my group with diffusion map embedding, and the code link for that is presented at the bottom. It's also available at the end. Okay, so that's the basic, that's one of the critical steps. We're not doing this on the connectivity graph, we're doing it on the graph Laplacian. One further point to mention about this is that in these really dense graphs, when you're doing a voxel by voxel analysis here, the results from doing a standard linear PCA and these fancy nonlinear embedding approaches, the results are actually quite similar, and this has to do from the density of the graph, and there have been some nice reviews that have covered these issues as well that we can discuss. But that is the basic approach. We're looking at variance in the graph space, and instead of trying to subdivide, we're simply looking at the embedding of that space in this low-dimensional representation of it, and then that's what's getting projected back to the cortex. Yep. Does that more or less make sense as the main method here? Are there questions? because that's basically it. That's what we got right now as a method. I mean, what's fascinating about this as an approach, I think we've already brought up spectral clustering. This is essentially a step less than spectral clustering. It's the spectral embedding, but without the k-means clustering part, where we cut this into different portions. It's simply saying, what is the maximum variance in this connectivity matrix, and how can we, if we ask the right kind of question, if we choose the right region of interest, how can we use that then to represent variance in that connectivity space, which corresponds to variance in, let's say, the functions that are being represented across that cortical area. That's the basic idea. Is that, feel free to jump in. We have some wonderful experts in the audience, so. Oh, please. Yeah. Absolutely. Absolutely. And I think that is one of the... So, yes, if you put in noise, you can get some wonderful structures out in the noise. That's true. And I think that's something that we've discussed with respect to all of these approaches, I would say. I'll get to that at the very end in some of the really important points for consideration. But it's absolutely critical. I think some of the wonderful work that I presented here from Hock and others, what's so important about the approach they take is that it's driven by a very strong hypothesis. It's validated against either task-based studies or other approaches, and that it's very well motivated in terms of research question, because you can absolutely get out these patterns no matter what data is put in. The method is going to drive the result in that way. One of the final, any other questions or points? Great. Okay. So the final step, and I think one of the challenges, is in spatial statistics that can be used. What Hock and colleagues have developed for this is an application of spatial statistics called trend surface analysis, which is a class of tools essentially for looking at linear or even nonlinear relationships over space. So it gives an approximation of how an individual gradient fits either a linear or group-derived spatial pattern. I put a link for a nice introduction to this topic at the bottom, and all the tools for conducting this, as I mentioned, have recently been made available online. The team recently put out a paper looking at how these gradients within the striatum relate to differences across individuals in behavior, and I recommend the link at the bottom as a further discussion of that topic and methods for addressing these kinds of questions. But fundamentally, I guess the question that we're asking is how do changes along a gradient, how can we look at differences across individuals in changes along a gradient? And that's one of the topics that has also come up, is how do we look at differences across parcellations across individuals? And this is a class of methods that are trying to address that by modeling the sharpness of that gradient and the fit of that gradient to a group model in space. Here is the code that just went up yesterday. I should have put the date. It would have been fun. But as I mentioned, there'll be a nice talk by Christian Beckman on this code this Thursday at 12:30 in the Open Science Room. The method's called Congrads for connectivity gradients. And it includes all of these steps that I've mentioned, including the surface trend modeling. I had to mention as well also that this is applicable to looking at large-scale gradients as well. So I focused on topographic maps within specific regions, but myself and others have looked at sort of large-scale overarching spatial patterns in connectivity. And on the left is a recent example of mine from 2016, and on the right is work that was just put out in bioRxiv by Gul and Satra and colleagues at MIT, looking at the cerebellum. And it's an example of how one can use these methods to ask the question of how different connectivity patterns relate to each other over the space of the cortex, and what is the spatial distribution of these patterns that we observe. It's one example among many. It allows us to look for degree of transition, the sharpness of boundaries. It opens up another set of questions that we can ask in terms of how we distinguish between connectivity patterns across the space of the cortex. And in this specific set of examples, I was using diffusion map embedding, and the example on the right was also using that approach. So in summary, and I think this is really critical, is like the selection of the analytic technique we choose is obviously going to result in a set of images Those might be sharply bounded clusters. Those might be really nice, smooth gradients. But it's the analytic choice, as we're all aware, that's going to drive that. And I just put this up there as clustering begets clusters, spectral embedding begets gradients, and it's kind of, you got to know what you want, which is a very difficult thing in this line of work sometimes when we have all of these wonderful tools available to us. No doubt that topographic gradients are an organizing feature of the cortex, but not all these gradients are meaningful. And it's absolutely critical to have strong hypotheses driving these approaches, because you can put data in and you can get gradients out, and if you don't have a strong hypothesis, it's really going to become challenging work to validate whatever results might come out of that. But these are useful methods for looking at trends in connectivity patterns or any other feature of the cortex across space at various scales. One can look at transitions between specific regions, one can look at changes within areas, and one can also look at these large-scale patterns and how they vary across connectivity patterns across the cortex. Some resources, I've mentioned it several times, but I can't thank them enough, the group at Donders for making their methods available as of yesterday. So now is the moment to get your hands on it. It's hot on GitHub right now. It really is. I think it's an exciting, when these methods finally become available, all sorts of fun things to try. And that, there's the link, Thursday, 12:30 in the Open Science Room with Christian Beckman. Diffusion map embedding, which has been on GitHub for a bit longer, and it's the one that we've been using in my group, is available through Satra's GitHub account. There's the repo, map align, and some examples of how to use that are available at this URL here. And of course, NiLearn has numerous methods based on scikit-learn for embedding of connectivity data implemented in Python that I can highly recommend. Thank you for your attention. Happy for any questions.